DNA in an organism's genome contains instructions that determine that organism's traits and coordinate the growth and development of that organism. Genes influence traits. Similar to a recipe can be changed slightly and change the outcome of that recipe. So that also happens with genes, that genes can be changed slightly, and that will change the outcome of a trait or the growth or the development of that organism. Corn is an example of changing genes to change the traits of a corn. Now, corn has been genetically modified both artificially through genetic engineering and also naturally through selective breeding. Changes in any section of DNA are known as mutations. So with selective breeding, this is a 9,000-year process that Native Americans developed over time from a wild grass called teosint that has no ears, no cobs, and no kernels. Well, over 9,000 years, Native Americans developed this by selective breeding, selecting for the wild grass that had some cobs and larger, eventually getting into what's known as maize. So this is natural, selecting for the size of the cobs, the size of the kernels, and for crop yields due to natural mutations that occurred in the genes, in the DNA. Now this is a major source of food. What began as a wild grass growing randomly all over became maize that is cultivated, and what we have in large-scale industrial farming now is corn that grows in narrow rows close together. Scientists have additionally artificially modified the genes of corn in order to have corn produce its own insecticide. So if you've ever opened a corn and found these worms that are corn borer worms that burrow in underneath the corn husks and eat this corn before consumers can get to it. The genetically modified corn can resist this worm by producing its own insecticide in the corn. We know that corn is susceptible to this European corn borer. So that's an infection that happens using genetic engineering techniques. This is a type of corn, this GMO corn, that is resistant to this worm because it produces its own chemicals. The development involved taking the genes from a bacteria, BT for short, just snipping it out of the bacterial genome and then inserting it into the corn genome. Now you have a recombinant DNA. Just like the human insulin gene was inserted into the bacterial genome, now what you have is bacteria gene inserted into the corn genome that allows for that corn to now be resistant to insects because it produces its own insecticide and kills off the worm as it's eating the corn. The problem is you now have a corn that is genetically modified, and there are issues with that. Monsanto also developed a Roundup Ready corn, which allows use of a chemical called Roundup that kills weeds, but not the corn plant. And that's because the corn has been genetically modified to resist that specific chemical produced by Monsanto called Roundup. There are political, social, environmental, and economic issues with GMO corn, but we'll just look at the chemistry. What exactly is modified when there's a genetically modified organism? Well, it's the gene that is modified, and genes are specific sections of DNA within one organism's genome. This is how this all fits into this human organism. A human has their traits and development and metabolism. That's run by 20 to 25,000 proteins. Each one of those proteins or hormones or molecules has instructions to code for it, which is the gene. So going back to here, a gene codes for one individual protein. A gene is a section of DNA in the genome, and all of an individual's genome is packaged in chromosomes. Humans have 46 compact structures. They're called chromosomes, and chromosomes are located in the nucleus of a biological cell. Atoms have a nucleus, that's in chemistry, but in biology, cells also have a nucleus. So organisms are made of cells, millions of cells. Each cell has a complete set of information to make the whole organism, and that's the genome. This information is carried on those chromosomes, which are stored in every cell, and though every cell doesn't necessarily express all of the genes, it's important that every cell has the information. So your skin cell, for instance, has all the information to make the whole organism, but only the skin is what's made in the skin cell. Or an eye cell. An eye cell has all the information. What's expressed are the proteins necessary for vision. Chromosomes in the nucleus contain genes made out of DNA, and all the information to make each cell is stored on DNA, and that storage is a special template. So that's a special code as well as a special structure. So DNA has this double helix structure, which we'll look at the details of, and that DNA is then wound around proteins and coiled in the process, and then packaged at another level into a chromosome structure, and then all of those chromosomes, all 46 of them, are packaged into the nucleus of the cell. All the information to make that one organism is a lot of information. A lot of information is stored on these chromosomes. Imagine the human genome scaled up in size, about 1,000,000. Scaling up the genome so that it is 1,000,000-fold or 10 to the 6 times larger. At that size, each chromosome is about as wide as a ramen noodle. Then if you uncurl the chromosome and basically uncurl those ramen noodles and laid those noodles end to end, all 46 of those chromosomes would stretch from New York to Kansas. So that's how much physical DNA there is. But taking all of that ramen noodle and curling it up and folding it up inside the nucleus, well, still at the size of the ramen noodle, that would fit inside of a house. So that's the level of organization of all of this DNA.